So for this lecture, we are going to talk about the structure of nucleic acids. And so nucleic acids, of course, are going to be RNA and DNA. And in our pathway to discussing those structures, we have to discuss how they're built, what makes up those structures. And so we'll talk about the general structures of our nucleic acids and with respect to our common bases. And then we'll talk about what the difference between a nucleoside and nucleotide is, which is very important in the actual structure of the nucleic acid versus those nucleosides that are going to be used for energy, such as ATP. And then we'll talk about the bond formations that make up our RNA and DNA and how the RNA-DNA structure is. So in general, there are three basic components of a nucleotide. So every nucleotide is going to have some sort of nitrogen base. And the nitrogen base is really going to be what stores the knowledge, what stores the information. And of course, it is going to contain nitrogen. And those bases are going to be bound to the anomeric carbon or the carbon 1 on the furanose 5-carbon sugar. And so it is going to be a pentose sugar, 5 carbons. It's either going to be deoxy or ribose or deoxyribose. And of course, this all is going to be a part of this carbon number 2. And then you're going to have phosphates attached to that 5 carbon number 5 carbon. Without a phosphate, this structure here is called a nucleoside. Now, nucleotides are building blocks of nucleic acids. So a nucleotide plus N nucleotides equal RNA or DNA. And so they are, of course, going to be very important in transmission and storage of biological information, which is the function of DNA. RNA is going to have a much broader function, so there's going to be different classes of RNA, which we'll talk about, are ribosomal RNA, messenger RNA, and transfer RNAs. In addition, RNA can actually be enzymes. So ribozymes are types of RNA. Now we have our nitrogen bases, and in our nitrogen bases we have 2 parent compounds. We have our pyrimidines, which are a single ring structure, and our purines, which are our 2-ring structure. And you can see that adenine is a purine, and adenine, of course, is very famous for ATP. So it is adenine with its 2-ring structure that is going to be part of most of our energy requiring reactions in our bodies. Guanine is a 2-ring structure also. It's another purine. And guanine is another nitrogen base that is part of energy. So we have a lot of reactions that require G proteins. And so that requires GTP, which is going to be guanine. So our pyrimidines are going to be single ring structures. And in our DNA, we have cytosine and thymine. And of course, in RNA, thymine is replaced by uracil. And so for our thymine to become uracil, we are going to lose a carbon on our thymine to form that uracil. Now, our base pairing is going to, of course, pair up a pyrimidine with a purine. So here we have our adenine, which is always going to bind to thymine in DNA, or it's going to bind to uracil in RNA. And these are going to have 2 hydrogen bonds between them. And then we have cytosine, which is going to bind to guanine always, and these will have 3 hydrogen bonds in between the 2 of them. And so alternative base pairing with respect to our purines and pyrimidines is what really makes up the structure or the shape of the DNA or RNA molecules. Just a little bit about nomenclature. So we have our bases, which are those nitrogen bases, adenine, guanine, cytosine, and thymine. and then uracil RNA. We also have hypoxanthine, and hypoxanthine is going to be a base that's not encoded or not found in our DNA, but it's going to be a byproduct of our guanine and adenine, our purine bases. So as the guanine and adenine get broken down and recycled, hypoxanthine is an intermediate. And the nucleoside equivalence for hypoxanthine is inosine. And this is an example of inosine here. And so hypoxanthine or inosine is a very important step in our recycling of our purine bases. Now, the ribose or the deoxyribose is a 5-carbon sugar, and it's in the beta-furanose form. It's in a circle form, cyclic form. And we are, of course, going to have our anomeric carbon. And this is where we're going to eventually have our nitrogen base attached to. So that will be attached up here. So each hydroxide group in our ribose or deoxyribose has a function. So number 1, our anomeric carbon is going to bind to that nitrogen base. Our number 2 OH is what distinguishes it from ribose or deoxyribose. So deoxyribose would not have an OH group here. We also have in our 3 position, we are going to bind to a phosphate. So this is going to bind to a phosphate that will then link to another sugar, which will be the 5-carbon sugar of sugar 2, or we'll call this sugar 3. And then our 4th is going to close the ring, and our 5th hydroxide is going to bind to another phosphate, which is going to bind to the 3' OH of our sugar. We'll call this sugar 1, and this is sugar 2, and then this is sugar 3. So they kind of form a chain. So the 5' carbon OH is going to bind to a sugar, and the 3' carbon OH is going to bind to another sugar to start to form the nucleic acid. So it's important to know what the terminology is. So again, we have our base bound to the anomeric carbon. In this case, we have an OH here, so this means it is a ribose. And then we have here at our 5' end a phosphate that's bound. And this phosphate, when it gets bound to a nucleoside, becomes a nucleotide or a nucleoside monophosphate, AMP. So in other words, if this was going to build a DNA or RNA molecule, that nucleotide phosphate would not bind to another phosphate. It would bind to another sugar. But if it was going to be part of our energy, ATP, AMP, ADP, then that is called a nucleoside, and it can bind to another phosphate. So it can bind to another phosphate, and it can become ADP, and it can bind to another phosphate and become ATP, nucleoside triphosphate. So a nucleotide has only one phosphate, and nucleosides can have 1, 2, or 3 phosphates. And they all have, of course, a base, and they all are attached to the sugar. Now, with respect to our phosphodiester bonds, we are going to basically have a linkage of nucleotides to form the nucleic acid. So you link and of course that is DNA or RNA. And so our 5' phosphate of one nucleotide is going to be joined to the 3' hydroxyl of the next nucleotide. So here we have our 5' phosphate, and it's going to be attached to our 3'. And this is called a phosphodiester linkage. Okay, I don't have to put that there. The backbone of our DNA refers to the alternating phosphate and pentose residues. And the bases are the side groups that are joined to the backbone. And these, of course, are going to be joined at regular intervals because they're always going to be on that anomeric carbon position. So they're always bound to those anomeric carbons. Now, this is going to have a 5' to 3' orientation. And this is going to give the strand its polarity. Now, the 5' end is going to lack a nucleotide at the 5' position. So the 5' end does not have a nucleotide. The 3' end is going to lack a nucleotide at the 3' position. The 5' to 3' orientation refers to the ends of the strands, not to the individual orientation of the phosphodiester bonds linking the nucleotides. So it's always, this is 5' to, I can make that better. Three prime. Okay, so that's the orientation. Now, key to convention is that when you are writing a single strand of nucleic acid, you always write with the 5' end at the left and the 3' end at the right. So this would be 5' to 3'. And then you would give the sequence. So it is 5' to 3' direction. Now, a short nucleic acid of about 50 nucleotides or less and this is going to be a oligonucleotide. And if you have longer nucleotides, so greater than 50, this is going to be a polynucleotide. All right, so in 1953, we know famously that Watson and Crick discovered the DNA structure. And originally, Watson and Crick had thought that the structure had 10 base pairs in each turn. And in reality, it is actually 10.5 to make it a very nice uniform structure that we know. Rosalind Franklin and Maurice Wilkins were the ones that initially took pictures using X-ray diffraction and were analyzing DNA fibers. And they found that this created a helix type of structure. And this X-ray diffraction was used by Watson and Crick to propose their model for DNA. Now, DNA is, of course, a double helix. The pairs are purine pyrimidine, and they are equidistant throughout. So you have equal distance between paired bases. You always have a purine with a pyrimidine, even though it might be AT and GC, and because of that, you have equidistance. They're going to be twisted at the top and bottom. So they get twisted at the top and bottom, and they're going to have a hydrogen bond that is going to give them what's called a stacking force. So they hydrogen bonded base pairs. Now the hydrogen of the third carbon has an oxygen group and its phosphate is going to dissociate at a physiologic pH. So that's at the 3' end. In addition, you're going to have what's called major and minor grooves that are going to alternate. And the grooves are going to be exposed and they're going to be able to interact with proteins. Now, there are many proteins that are going to interact with DNA. We often think of our polymerases, of course, such as our polymerases. But there's also going to be transcription factors. We're going to have enhancers. We're going to have primers, etc. Now, one strand of the DNA is always going to serve as the template for synthesis of the other. And of course, the DNA strands are going to be referred to as anti-parallel. So you have, for example, 5' to 3', and then you'd have on the other end 3' to 5'. So they're anti-parallel. The two antiparallel chains are going to not be identical in sequence or composition, but they are going to be complementary. Let me see. So, for example, adenine in one chain correlates with thymine in the other, etc. The complementarity is attributable to the hydrogen bonding between the pairs, and the hydrogen bonding and the complementary make major contributions to the stability of the helix. So hydrogen bonding and complementary are equal stability. I'll write it up here. Now they are going to, of course, have an anti-parallel template, and that's shown here. So we're going to have, for example, our parental DNA double helix, and then we're going to have strand separation, where now your two parent strands are on the outside, and that's shown in blue. And so these two parent strands are the template strands. So this is template, and this is the template strand. And then you replicate and make the new daughter strands, the two new synthesized strands. And so, again, this has to go with the DNA strands are anti-parallel. So here, of course, your daughter will have a T, and here it will have an A, which is complementary to the parent strand, but they're the newly synthesized strands. So the template strand is very important in the fidelity. So fidelity of DNA is that you get past the same information from cell to cell, from parent to offspring. And the fidelity is what really makes or gives the ability of us to transfer our information, our DNA information, onto generations throughout time. Now, finally, there are a few unusual structures of DNA that we can talk about. We have our palindrome sequences, and a palindrome is essentially a sentence that is spelled identically, read either forwards or backwards. So palindromes are going to be the same forward or backward. And so these are going to be in the DNA that are inverted repeats. So we'll put 1 here and 2 here. So 1 and 2 are inverted repeats. Now the palindrome sequences are going to be complementary, and they are going to be able to form 2 structures. The first structure they can form is going to be called a hairpin. Oops. So we'll put A here. A is a hairpin. Looks like a hairpin. And B over here is a cruciform. This looks like a cross. Now, when you have a single DNA or RNA strand involved, you get a hairpin. And so this is our strand, our 5' to 3'. And these 2 are complementary, so they kind of fold onto each other, and you get this hairpin. And this can be used for regulation, for example. If you have 2 strands involved, you then get the strands that are going to be complementary to bind to each other. So here's our strand 1, and here's our strand 2, and they form a cross structure. Now, these again are going to form, and they're going to be used in a lot of bacteria or smaller genomes to control gene transcription. Eukaryotes have a lot of different ways to control transcription, but if these are present, then it makes it so that, for example, your DNA polymerase cannot continue, your RNA polymerase cannot continue, because it doesn't know how to deal with these hairpins or cross-shaped structures. Okay, so that's what I just said, and that's the end of it.